Assalamualaikum warahmatullahi wabarakatuh. My name is Luqman Hakim bin Ruzman. I will be presenting my final year project entitled Exploring the Diversity of Slipper Gobies in Penang region using an Integrative Taxonomic Approach. Overview. This study focused on the diversity of a fish group from family Butidae. Until recently, only few studies have ever been done on this family in Penang region, and thus I re-examined their taxonomy by utilizing a modern or integrative taxonomic method that combines both morphology and molecular taxonomy. This study will prove the efficiency of using integrative taxonomy approach in order to accurately determine the diversity of a fish species Butidae in Penang region. Butidae or butates are widely distributed and they are urinaline, which means it can be found in fresh, brackish or saltwater. Although it is widely distributed, the accurate number of species that can be found in Penang region is still not clear. It is also underestimated to be only 3 species present in Penang region. In order to discover their diversity, modern taxonomy utilizes molecular markers which use DNA fragments combined with morphological evidence is used to accurately determine or update the identity of family Butidae. Hence, taxonomists usually use cytochrome C oxidase 1 gene from mitochondrial gene, or we call it CO1 gene, as a DNA barcode to differentiate each species. This gene is known to be widely used in fish taxonomy studies. The objective of this study is to accurately discover the diversity of Butidae in Penang region. The second objective is to be able to differentiate each species based on their morphological and molecular characteristics. I also aim to build a specimen-based reference for this fish family at School of Biological Science, USM. It is hypothesized that by applying integrative taxonomy method, the taxonomic diversity of Butidae in Penang region can be accurately discovered or resolved and be used for future research. The methodology used in this study is quite simple and consists of 2 parts. The first part focusing on delimitate or differentiate the specimens into groups, or we call it operational taxonomic units OTU, based on their morphological and molecular characteristics. The second part will be identifying or naming the specimens by comparing them with the reference database available and previous journals. The results will be then compared with GenBank database, and specimens that are hypothesized to be misidentified by previous researchers will be re-identified based on the current study findings. Lastly, the results of this study will update the diversity of fish family Butidae, which can be used for future reference. To describe the morphological variation among specimens of Butidae, I took 19 morphometric measurements and 11 meristic counts, which follows previous research methodology. For molecular examination, it involves DNA extraction and amplification of targeted CO1 gene using polymerase chain reaction or PCR method. The sequence obtained will be analyzed and aligned in order to observe the molecular variation between the specimen which shows intraspecific and interspecific variation. Maximum likelihood tree is then built, and automatic species delimitation method is used as the comparative methods for the results to compare between each specimen and their variation. There are 2 sampling sites used in this study. The first sampling site is in Sungai Merbuk, Kedah, which illustrated with yellow point. Main vegetation at Sungai Merbuk is mangrove plants, which is the natural habitat for butids. Next site is in Sungai Betung, Penang, which illustrated in red point. Figure 2 shows the equipment used to capture the fish specimens, which is a large traditional fishing traps that are called pompang, operated by local fishermen. Figure 3 shows the software commonly used by molecular taxonomists in order to align the sequence obtained from the specimen, which is MEGA7 software. Morphological results obtained from 16 specimens that were caught is that the specimens were differentiated or delimited into 6 OTUs. OTU-F are not shown because no preliminary examination can be done on the specimens due to COVID-19 lockdown restrictions. The specimens were differentiated by comparing their morphometric measurements, meristic counts, as well as their body shape and coloration. Thus, based on the morphological examination, 6 species were hypothesized to present among the specimens obtained. Next, molecular examination is done by building a maximum likelihood tree of the specimens, shows that there are 6 different species among the specimens. The length of the branch also shows that the specimens are clearly differentiated into their own OTU. This means that there are possibly 6 species present based on the tree. Results from ASAP software at the right shows that the best partition are the one with the lowest ASAP score, which is 5, with 23 number of species present in the dataset. This dataset includes the sequence of Butidae from Southeast Asia region obtained from GenBank. And among the 23 species, the 16 specimens used in this study are also delimited into 6 different species, which shows the same with the maximum likelihood tree. Thus, each morphology-based OTU obtained from morphological examination are fully supported by molecular results, which using CEO1 gene sequences. After confirming the number of species present among the specimens, the next part will be identifying each OTUs by comparing their morphological characteristic with previous research. Each OTU has their own characteristic which differentiate them with other species. For example, OTU A and B are identified as Butidae humerilis and Butidae butiis respectively based on their maxillary length characteristic. OTU C and D are identified as their own species based on their cheek pattern and body shape that is different from other species. And presence of spines on the first branchiostegal race of OTU E identifies it as Oceolotris europhthalmoides, and OTU F are similar to OTU B, except that no auxiliary scales and scale row between eye and on interorbital ridge of OTU F can be found, and that identify OTU F as Butidae amboinensis. Morphological and molecular results obtained will be compared with previous study in order to resolve or update the diversity of Butidae in Penang region. Interestingly, the diversity of Butidae has increased from 3 to 6 species present in the Penang region. The presence of Butidae amboinensis was expected because these species have been collected throughout the west coast of Peninsular Malaysia. However, the presence of Prionobutis microbes and Oceolotris europhthalmoides were unexpected because it is the first record of both species in Penang region, and it is also the first record of both species in Peninsular Malaysia. Prionobutis microbes presence was more surprising because these species are known previously to be only distributed in Australia, Papua New Guinea region. So further study on Prionobutis microbes is needed to be done in order to discover the source of introduction of this species in Penang region. In conclusion, the objectives and hypothesis of this study is met. The taxonomic diversity of Butidae is revealed in this study with the increasing number of species present from 3 to 6. All 6 species also discovered in this study are successfully named. The third objective is met by adding 15 new specimens which obtained from this study into USM fish collection to build a specimen-based reference collection for further studies or for future references. The hypothesis of this study is accepted, which is by using an integrative taxonomic approach method combining morphological and molecular taxonomic, the taxonomy of fish family Butidae in Penang region can be resolved. In a nutshell, future research can use this study as references in order to study the diversity of Butidae, because the findings of this study are reliable since two methods were used and detailed information to identify species of Butidae are also provided. However, the limitation to this study is that small taxonomic samples were used in this study. This can be fixed by increasing the number of samples in order to accurately discover the diversity of Butidae in Penang region. Finally, this study has successfully proved the efficiency of using integrative taxonomic approach in order to resolve the diversity of Butidae in Penang region. More studies on this family in Penang region are encouraged to be done, especially with the presence of first recorded species of Butis amboinensis, Prionobutis microbes, and Ocelotres europhthalmoides in Malaysia. Last but not least, I want to thank my final year project supervisor, Dr. Sebastien Lavoue, for his help and guidance in order for me to complete my final year project. I would also like to thank School of Biological Science for giving me the opportunity to work in a lab, as well as lending their equipment in order for me to complete this final year project. Thank you.